In this session, I'm going to talk about DNA extraction procedure in banana cells. In order to extract DNA from banana cells, we have to take an organ from banana, break down the organ into small pieces of tissues, and then treat the tissues with certain chemicals so that we break down the cells into a level that we can take out the chromosomes. To achieve this goal, we have to understand the basics of DNA extraction, especially genomic DNA extraction, and then the role of certain chemicals in the procedure, and then go through the stepwise procedure. DNA extraction is a process of taking out DNA from an organism's cells. The process would include taking an organ, and this is followed by tissue breakage and grinding them, and then finally treating the cells and the tissues with certain chemicals to take out the DNA. As you can see the steps here, we chopped the organ into small pieces, we weighed them, and then prepare the lysis solution, and finally separate the DNA afterwards. This looks simple, and it is quite simple actually. The chemicals to use include using sodium dodecyl sulfate, which is SDS for short, and warm water, distilled water. We preheat the water and then use this to treat our broken down tissues, the banana tissues. And then we use NaCl as well as EDTA. The NaCl is useful to do certain things. So the first step included SDS to break down the cell membrane. The second step included NaCl to take out the cell sap and affect certain proteins. The EDTA is a chelating agent that gets the metallic ions like magnesium and hence stops the activity of certain enzymes like DNase enzymes and prevents the DNase enzymes from breaking our genomic DNA. After that, we use chloroform in order to denature proteins and change the shape of the proteins. These three ingredients all together, when we use them against the tissues that we have, we get a cell lysate. And upon precipitating the cell lysate, we would have DNA in solution and the other unwanted materials like proteins and lipids as precipitated materials. Then we can use ethanol, absolute ethanol let's say, to extract the DNA in this way. So the first step is to weigh 100 grams of banana and then weigh SDS. Similarly, we can weigh the other materials like the NaCl and then prepare EDTA. So after that we can grind the banana using a grinder or juicer. So these are the ingredients. We weighed 100 grams of banana and put 250 milliliters of preheated distilled water. After that we used 10.5 grams of NaCl and 3.5 grams of SDS as well as 3.5 milliliters of EDTA, and we use that in the mixture. This is a mixture that breaks down the cells and also forms foam, which is unwanted. To get rid of the foam or the majority of foam, we can filter the cell lysate with a gauze. As you can see here, we can get rid of the majority of the foam that has been formed because of the SDS. You can also use isoamyl alcohol to prevent foam formation. And after that, you can separate the cell lysate into certain tubes and adjust the volumes and then centrifuge them at this RPM, which is maximum RPM of our centrifuge here. After that, we transfer the supernatant to clean tubes and then add few drops of ethanol and finally observe the results. When you add ethanol, you would get the chromosomes shrunk together like this and the ethanol surrounding it. You can then take out this chromosome into a clean tube. You can wash it with distilled water or precipitate it first by 70% of ethanol and finally wash it 2 times with distilled water and store it for later use. And that's how you extract chromosomal DNA from banana cells. Thank you.